This is IAQ Radio, Indoor Air Quality Radio, the voice of the indoor air quality industry, with your hosts, Radio Joe Hughes and the Z-Man, Cliff Zlotnick. And now, Radio Joe Hughes.
Good day and welcome to IAQ Radio Plus. It's episode 736. We're back from vacation. I may be a little rusty, but I'll do my best. This week, we welcome Dr. Tess Eidem from the University of Colorado Boulder to talk about IEQ, bioaerosols, and worker health in cannabis manufacturing, a fast-growing area, Learn more this week on IAQ Radio Plus. Before we get started, let's thank our sponsors. They're the reason we can continue doing the show. Don't forget, after the show, you can continue the discussion on afterthoughts.iaqradio.com. IAQ Radio Association sponsors are AIHA, the American Industrial Hygiene Association, at aiha.org. IICRC, the Institute for Inspection, Cleaning, and Restoration Certification, at iicrc.org. The Restoration Industry Association, RIA, at restorationindustry.org. The Environmental Information Association, EIA, at eia-usa.org. Actionable Insights at getinsights.org. IAQ Radio industry sponsors are Particles Plus at particlesplus.com, BioPlanet at byoplanet.com.
And now you can win a cool prize. It's time for the IAQ Radio Trivia question. Be the first to correctly answer. Simply email your answer to czlotnick@cs.com. Or if listening live, just text your answer from your computer. And now, here's the Z-Man.
Hello, everyone. Here's today's IAQ Radio Trivia question. Name the scientist who was first to systematically study living and fossil single-cell organisms with microscopy, and who began what is today the oldest major collection of micropaleontology in the world. Back to you, Joe.
All right, Cliff. So Tess Eidem's doctoral research was focused on developing novel antibiotics against bacterial pathogens. I'm going to be learning more about that here soon. Her scientific career is centered on bringing modern microbiology and RNA biology practices to promote human health. Leveraging her scientific expertise, she transitioned into cannabis industry, where she spent 4 years working directly in cannabis production and quality management. After witnessing firsthand the challenges of microbial contamination in the cannabis industry, Dr. Eidem returned to the University of Colorado Boulder, where she currently works as a senior research scientist in the aerobiology and disinfection laboratory. Welcome, Dr. Eidem. Great to finally get to meet.
It's awesome to be here. Very excited to chat with you guys.
All right, so are you in the lab now? That looks like a lab background.
Yeah,
to me.
you can tell. Yeah, we've got all the assorted hodgepodge scientific supplies, and not in any particular order.
So, going back to your very brief bio we did there, what in the world is developing novel antibiotics against bacterial pathogens? Tell us a little more about that.
Yeah. So, bacterial microorganisms are big pathogens. They infect people. They can cause all sorts of disease, from lung diseases, skin, blood diseases, heart diseases. And the number of antibiotics that are effective against them is slowly diminishing because, microbes are actually really good at adapting and becoming resistant. And so, we as scientists are always trying to increase the arsenal of antimicrobial agents against these bacterial pathogens. And when you look at what's clinically available, there's actually just a handful of different types of antibiotics out there. And if you were to think of a microbe as like having essential organs or essential pathways, like if I shoot you in the arm, it's probably not going to kill you. But if I shoot you in the heart, then it could. And so there are these essential pathways within the bacterial cell that you want to target. And like I said, current clinically available antibiotics only target a handful of them, but there are hundreds that you could potentially target. And so we were looking at the RNA processing and decay pathways. So once an organism, it'll take its DNA, make it into RNA. So this is part of the central dogma. So if we have any biologists here, they'll know. So your DNA is your blueprint. It has all the genes that you need to make everything that you need to function. You then take parts of that DNA, make it into RNA, and then that is translated into protein. And that protein is mostly a lot of functional stuff. And a lot of things will target DNA replication. It'll target protein synthesis or protein function. But there's this big gap in the middle with RNA metabolism that's not targeted. And it's pretty different in bacteria from human cells. So that makes it a good target for antibiotic drug discovery. And so that's what we were doing. We were developing small molecule inhibitors of different RNases, which are, not all of them are essential in bacteria, but in gram-positive bacteria, RNPA, which was my favorite RNase, was. And so we were developing these small molecule inhibitors, and we showed that not only did we block those RNA synthesis or mostly processing and degradation pathways. I'm getting a little technical here. But we blocked it in the cell, and then we also inhibited growth. And we could basically show that these molecules basically present a novel potential class of new antibiotics. And that's what my PhD work was all focused on.
And I assume people are still working on this. You've
Yes.
switched over, but
yes, yep. My old boss, Dr. Paul Dunman, is still working on developing novel antibiotics, really focusing on these RNA pathways. And there are papers that have come out after me working with medicinal chemists to improve on the structures of those small molecule inhibitors to increase their efficacy. So yeah, it's still ongoing. Antibiotic drug discovery takes a long time, but
yeah,
it's on its way.
I just haven't seen anything new. It seems like they're working on it, but they're not there yet.
Yeah. A lot of bacterial pathogens are resistant. They're becoming resistant. Staphylococcus aureus was the microbe that I mostly focused on, but there's something called the escape pathogens, which is an acronym that stands for E. coli, Staphylococcus aureus, Klebsiella pneumoniae, oh man, Acinetobacter, man, this has been a while. I haven't talked about this. Acinetobacter baumannii, Pseudomonas aeruginosa, and other Enterobacter species. But then, on the mold front or on the fungal infection front, which I'm definitely getting more involved in now, and also with my work in cannabis, lots of fungi are getting resistant to different antibiotics. And so there is a call from the World Health Organization to focus on these critical priority fungal pathogens, not only because they cause disease, but because the number of effective treatments are kind of dwindling to, once you get infected, it's much harder to recover from having those types of infections.
Interesting. Very interesting. Okay. Now you're now with the aerobiology and disinfection laboratory, which I can kind of guess at what you do, but maybe you could tell our audience a little more what you do at this particular laboratory.
Yeah, so I work with Dr. Mark Hernandez, and
a friend of the show. Hello, Mark.
Yep. He was bummed he couldn't be here today. And yeah, so our lab does a lot. We have actually a really diverse kind of portfolio of different projects, but a lot of the aerobiology stuff that we work on is focused on different airborne bioaerosols. So things, biologically derived components that are floating around in the air, whether it's viruses, bacteria and bacterial spores, fungal organisms, fungal spores. And now I really work a lot on aeroallergens, or those airborne allergens that float around in the air and cause harm through the route of inhalation. And we do
right now.
Yeah. If you're allergic to pollen or grasses, you probably are feeling it. Or if you've ever experienced it with cats or dogs or dust mites, things like that, all of those are big triggers for allergies and even asthma and asthma attacks. And so that's really why, allergies can be annoying. And a lot of people are like, oh, this is like slightly decreasing the quality of my life. But for some people, allergies can be really severe, even leading to anaphylactic shock. And so it's really important to pioneer this area to look specifically at aeroallergens, because most research in this area don't like, it doesn't really focus on looking at the allergens themselves. It looks at like spore counts or some surrogate, or it'll collect dust and then look at allergens or spores or other types of surrogates in that dust, but they're not actually, very few researchers are actually looking at the actual allergen in the air. And so that's what we're doing here. We do it in controlled chamber environments. And then we will be processing, we're incorporating this into some of our work in Denver public schools. So we're in several hundred classrooms monitoring air quality in Denver public schools. This isn't a project I directly work on, but it's another project in the lab. And we monitor like particulate matter in those spaces. We go in and collect bioaerosols, and then we put in, and then we swab desks for different types of pathogens, mostly viruses. And then we put in like HEPA filtration, and we kind of compare before and after and see what kind of intervention strategies can be useful, especially in these small spaces that sometimes might not be very well ventilated, things like that. So yeah, that's kind of a big overview. We do a lot of other stuff with surface sanitation and sterilization. And Mark also does a lot with wastewater treatment stuff too. So it's a wide diversity of projects that we work on in the lab.
Well, one I want to mention, because we're going to bring Mark back here as soon as possible, but he was working on a lot of HVAC cleaning type projects with NADCA, the National Air Duct Cleaners Association. Very interesting work. We will be talking to him about that. But let's move on to your cannabis work. Tell us a little bit about growing cannabis. Like, I don't think people, some grow it in their backyard, some grow it in their attic, some grow it, in these large grow rooms. Tell us a little bit about how you got involved with that.
Yeah, so I was a scientist for a long time, transitioned into a professor position, and ended up sort of finding myself not being a professor anymore. So I kind of left that space and was on the search for a new role, and moved back to Rochester, New York, where I went to graduate school, and got a job at Columbia Care, which is a multi-state operator. It's a cannabis manufacturer. And I was the laboratory manager there. So I oversaw all of the extraction. So taking that cannabis flower and extracting the cannabinoids and other oils out of it, and then purifying that. So kind of getting all the lipids out of there, all of the, kind of gunk and other like non-desirable things, and then purifying it through distillation. And then we also did a lot of formulation, and this was back when it was more of a medical market. So we made caps, we made tablets, we made suppositories, lotions, tinctures, more medical grade type of products for our medical patients in New York. And then in 2020, the sale of flower started to be allowed. So that's the actual flower component, the herb, from the cannabis itself. And before that, you could not sell flower in New York State. And so we were the first to actually get flower to market in New York State, our group at Columbia Care. And, as we were getting flower up and running, it was pretty different from some of these medical grade sort of products that we had previously made for years and years. And so it took, we wanted to put in some preventive measures. What was really cool is we were working in the old Kodak Center. A lot of people in my group, which I was really fortunate with, like had master's degrees in horticulture, agriculture, and on my side, a lot of chemists, a lot of biotechnology folks. And a lot of them had food safety backgrounds, and I did too. And so we started to put in some preventive measures. We built a HACCP plan, started doing environmental monitoring. We started building in-house testing. So we would test our product for microbial contaminants before it went out for third-party testing. And it really was just a really cool experience to see this new challenge come to us. And we all rose to the occasion and started adopting some of these good practices in other industries so that we could put out, really good quality flower. And at the time, the regulations in New York were really, really strict. And so it was very, very challenging to get in New York. Like, for example, I'm in Colorado right now, and there are microbial thresholds on cannabis flower. There's total yeast and mold counts. So it's basically like your fungal populations can't exceed 10,000 colony forming units or CFU per gram of cannabis flower here in Colorado. But in New York at the time, it was 50. So compare 50 to 10,000. It was very, very strict. And they were using basically pharmaceutical levels for inhaled products to apply to a medicinal herbal product, which was definitely not in line. I'm all for, regulations and as long as they're reasonable and make sense. So now they've loosened those regulations, but at the time it was very challenging. Yeah.
That was my question. Have they changed that? No.
What's
their level now?
I think it's 100,000 CFU now, but they don't test, but it's funny because they actually don't fail product for CFU counts. So you can have like a failing level. But right now they're mostly testing for pathogens. So if you find like Aspergillus fumigatus, for example, then they'll actually fail the product. But otherwise in New York, they're testing for it. It's required to test for colony forming units for total yeast and mold, but they won't fail you if you're above a certain threshold. So it's like an interesting, weird spot that New York is in. That's not how most states do it.
So part of the problem is this is all regulated on the state level now, and there's no federal oversight of it. Am I accurate in saying that?
100%. There is no oversight from the FDA or USDA on cannabis products. And that is really challenging because, the states have these piecemeal regulations. Not everybody agrees. There's not a lot of data or funding to support research to really investigate the hazards to cannabis users and patients. Many of these folks are being prescribed cannabis for pain or, appetite, cancer patients, things like that. And they're usually immune compromised in some way. And so keeping that intended user in mind is really important when we're thinking about the consumer safety issues that folks can maybe be presented with when they're consuming these products. And yeah, it's very challenging because unlike food, where you have very clear like food safety guidelines, like Food Safety Modernization Act or FSMA, there's all sorts of I'm a Preventive Controls Qualified Individual or PCQI. You have to build a food safety plan and you have to check along each step of your process that you're, meeting certain safety requirements. That's not required in cannabis. And so what ends up happening is, it's that process that the FDA has in food is considered like a quality by design process where you're really building quality into the process and checking along the way. Cannabis is a quality by testing process where you're basically operating blind to your contaminants until the very end where the product is tested, and then it either passes and you can sell it to consumers or it fails. And depending on your failure, if it's microbial, many states allow microbially contaminated products to be recovered through decontamination methods like remediate. It's called remediation. So you just zap it with a radiation or ozone, and then you can sell it to consumers. But that product could be, we don't really know the hazards that having all those microbial skeletons all over that product could really pose to consumers.
Very interesting. Now, real quick too, the hemp is a big topic here in Pennsylvania, and I think in other states, allowing the growth of hemp, which is, well, if you could tell us the difference between hemp and what you worked with, cannabis.
Yeah, so hemp and cannabis are both the same species. They're just basically have been bred for different purposes. So hemp is typically bred for its fiber, its seed oil, and sometimes for CBD. So sometimes it also is bred for that. So CBD is a non-intoxicating cannabinoid. It's anti-inflammatory and has other potential, beneficial medical applications. And hemp is really defined as having less than 0.3% THC in it. And so that's kind of the USDA definition of hemp. But really, hemp and cannabis are both the same species. And so there's actually a lot of hemp growers who are growing high THC flower, and they're calling, in the flower form, it's its acid form. So it's THCA. And so they call it THCA hemp. But basically, if you were looking at USDA definitions, that is cannabis. So, and then hemp is not usually smoked. So it's not usually grown for smokable flower, although it can be. It's usually grown for, again, it's fiber, oils, hemp seed. And so it's mostly extracted for that CBD.
Hey, Joe, I was going to say in Pennsylvania, they have this limit of what the THC level can be. So apparently they go out and somebody goes out to the crop or whatever and they measure it.
Right.
And it cannot exceed that. I think if it exceeds that, then they have to destroy it or whatever. So the timing for the harvesting of this is really important. Who does that? Would the state would
usually
monitor?
The state will do that, or you'll send a sample out to state testing. Hemp can be sent across state lines, unlike cannabis, so you may elect to send it out of state as well. That happened to my friend. Her family has a farm in southern Colorado, and they grew hemp, and hemp is notorious for being hot its first year or two.
Right.
And so it exceeded that 0.3%. And like when you're in that spot as a hemp grower, it can be pretty devastating, because a lot of times you just have to destroy it. Or what some people do is just throw in a bunch of stems and leaves and stuff to try to bring the average THC level down. So there's ways people can like get around that, but a lot of people now are at least I see more and more evidence that folks are just like selling it as THCA hemp. So that's not going to go through cannabis regulations, even though it is in violation of that USDA. And there's really not a lot of teeth against that right now. So we'll see what ends up happening. There's a lot of Wild West going on in the cannabis space.
I'll bet. Now let me clarify something for myself. Hemp is almost always grown outdoors. Is that accurate?
Yes. Yeah.
And therefore we would be also concerned about allergens.
Yes, you could very well be. Some hemp is grown with both males and females outdoors. So you may find, especially if you want hemp seed, so cannabis plants have both male and female sexes in the species. Some plants will have both, on one plant, but there are two separate sexes for cannabis plants. And females, when they don't have a male around, will make really big, beautiful flower. But if a male is around, then it will fertilize the flower, and the flower will actually not grow that much, and the plant will dedicate more of its resources to making seed. And so for folks who want to make hemp seed, you will have both male and female plants around. And there's like all sorts of research, which is really cool, kind of like saying, how many males do you really need in these outdoor grows? But the males have, they release pollen, which we're talking about earlier in the show. Pollen is an airborne allergen. It has different protein allergens that are presented within the pollen granules themselves. And so pollen can be an allergen either in these outdoor grows or even just ditchweed, things like that, kind of these wild cannabis strains that are growing around. I grew up in Nebraska, definitely, along creeks and things like that. You see it. And so you can definitely develop allergies against cannabis through pollen exposure.
But that is not an issue with indoor grow where they are just using female plants, correct?
Yes. So this is something that I've seen in the literature. And I'm really glad you bring it up because, when we're talking about occupational exposures to cannabis, pollen is probably not going to be on the top of the list when it comes to being exposed to cannabis allergens. That route is not as likely in these indoor grows. Males are typically not present unless you have a breeding program or things like that. Sometimes females can, it's called herming out, so they can kind of hermaphrodite and produce not true like male parts, but they can produce some pollen. But typically it's not really an issue. You don't have pollen in these indoor grows.
Okay, so from your experience, the issues that do occur with worker health are not from the pollen on, unless you're outdoor, or obviously that could be a problem, but with indoor growth, not so much of an issue.
Right, right. It appears from an allergy, inflammation, inhalation hazard to bioaerosol perspective, it's much more likely to be, mold spores and other microbial metabolites that can be in the air, or ground cannabis dust that can be those inhalation hazards that workers are being exposed to.
Okay. All right. Now let's talk a little bit about, cannabis cultivation and indoor environmental quality in the big picture. Then we'll focus down more on bioaerosols. But in the big picture, what are we looking at as far as temperature, CO2, relative humidity, VOCs? What kind of indoor environment are they trying for?
Yeah. So I'll go through a little bit of the cannabis cultivation and manufacturing process. I have a figure of this in that paper. It's the figure 1, and it's pretty cool because it goes through the cultivation, dry cure, and the manufacturing side. Usually in a vertically integrated cannabis operation, which many of them are, you will have all of these operations happening either in the same building or very close together.
Okay. Grayson, can you put that up for us real quick, that figure 1? Go ahead. I'm sorry.
No, you're good. So in cannabis, most cannabis strains or like genotypes, they're propagated. Oh, I might be getting a little feedback. Okay, we're good. They're propagated clonally. So you take basically cuttings of a mother plant, and then you put them into a rooting hormone, and then you put them into some substrate, and then you allow them to grow. And then all of those clones are going to be the exact same as the mom. And at those beginning stages, you usually have them domed because they don't have roots yet. And so you want to keep high humidity in those spaces. You want to keep them warm. So you usually have little seedling trays you keep them on. And so in that seedling stage, or in the clone stage, it's really high humidity, warm temperatures. Then as you go into vegetation, which I don't have here, I've kind of combined all of this into cultivation. Vegetative stage is when you're really trying to get that plant to grow as quickly as possible. You want to keep, so you'll hear a lot of growers talk about VPD, or vapor pressure differential. And that's really locking in the humidity and the temperature so that you are getting good transpiration, or basically the movement of water and nutrients through that plant. You want to have that dialed in. And so you usually see grows like between like 75 and 85 degrees Fahrenheit, and around like 75% humidity in those vegetative stages. Then in flower, which you can kind of see, there's a flowering plant in this figure. The flower are those kind of like cone shape in between the leaves. Like cannabis flower isn't like your beautiful daisy or something like that. I still think they're beautiful, but they're kind of like in the leaves and on the stem of those plants. And in flower stages, you usually want to bring the humidity down a little bit, mostly because you don't want those flower nugs now, those inflorescens, to get too moist, because you have the risk of mold developing in them. And then as you harvest, so after you harvest, you want to move that flower into a dry cure space. So here you can see this is kind of like a typical hang dry situation. So you'll harvest the plant, depending on, your particular SOPs. You may hang them with the leaves on, you may take off the fan leaves, you may shuck, you may take all the flower off and let it dry on racks. But
is this a separate room?
Usually it's a separate room.
Because on the graphic, it kind of looks like the same room.
So it is a separate room and a separate environmental controls for that space. And so in that space, you want to remove the moisture from the plant in a controlled way without removing other volatiles like the terpenes, which give the aroma and kind of that sensory aspect of cannabis. And many people do, they'll smell cannabis and they'll be like, ooh, this smells good. If you lose all that, you dry too fast and too warm, you're gonna end up with a lower sensory, a lower terpene content flower. But you don't want to dry it too slow, because then that's when you get bacterial and fungal contaminants in post-harvest spoilage microorganisms that can also be pathogens. And so usually you'll see people say like 60-60, so 60 degrees Fahrenheit, 60% humidity. I haven't seen a ton of scientific evidence on that. There is a really cool paper by Dr. Zamir Punja that talks about contributors, like environmental contributors to total use and mold counts on cannabis flower. And he showed, basically, he took samples during the drying process and measured them for their total use and mold counts. And what he saw is early on, you see like mold levels kind of be steady. There's an increase because now that plant is dead. It can't defend itself against these post-harvest spoilage microorganisms. It's food, and then it goes back down once it dries. And then in the processing packaging, you have your typical kind of manufacturing environment there, where you want to have like, probably 50% humidity or less, and ambient temperatures. And this is where you see a lot of exposure to ground cannabis dust. So now that plant matter is dried, it's more easily aerosolized, and so it becomes airborne, especially if there's improper ventilation. I mean, I've seen grind rooms that's the size of a closet, and you go in there and there's just a layer of cannabis dust everywhere. And so making sure that there's appropriate like filtration, ventilation, things like that, or appropriate PPE in that step is another area where you could be exposed to bioaerosols, but also the environmental conditions play a role in that too.
Interesting. Before we go, we're going to go to halftime in a moment. But before we do, you kind of just changed my perception of things. I'm thinking they probably actually use more air conditioning because you don't want that temperature too high,
right?
Both for the growth and for the workers. I mean, they're working in these indoor environments with a lot of humidity, high temperature. So, okay, interesting. Very interesting. All right, we will be back with the second half of our interview. We've got Dr. Tess Eidem, and we're talking about cannabis cultivation. Our association sponsors are AIHA, Healthy Workplaces, A Healthier World, AIHA.org. The Environmental Information Association, EIA's multidisciplinary membership collects, generates, and disseminates information concerning environmental and occupational health hazards in the built environment at EIA-USA.org. The IICRC, a nonprofit standards development and certifying body for the cleaning and restoration industry, IICRC.org. The Restoration Industry Association, the oldest and largest nonprofit professional trade association dedicated to providing leadership and promoting best practices through advocacy, standards, and professional qualifications for the restoration industry at restorationindustry.org. Actionable insights, no more mistakes, no more missed line items in your Xactimate estimates at getinsights.org. Industry sponsors are Particles Plus, feature-rich particle counters and air quality instrumentation. Count on us, particlesplus.com. BioPlanet at byoplanet.com, improving human, animal, and environmental health with electrostatic spray technology and advanced chemistries at byoplanet.com. All right, we're back with the second half. Cliff, let me turn it over to you because I know you had a particular interest that you wanted to talk about.
Yeah, thanks, Joe. Tess, one of the areas that our listeners are particularly interested in is odors, and odors that would be found, during growing and so on and so forth. And, oftentimes, they may encounter an illicit grow room or something like that, and then they have to deal with it. Also, sometimes the odors get out, they escape the facility, whether that's intentional or not, and neighbors and people like that can get involved with it. And then, sometimes, I guess recreational smoking is permitted, and then sometimes there are odors left after smoking, and someone moves out of a home or an apartment or something like that, and they're troubled by it. But just what can you tell us about odors and odor control, and any recommendations?
Yeah, odors are definitely, from a complaints, a lot of neighbors and people who are close to cannabis grows. Like, I mean, I can drive by, there's a couple of grows like not too far from my house. And if I drive by, I'm like, yep, that's weed. Like, I'm a couple blocks away, but I can smell that.
It's pretty distinctive.
It's very distinctive. Yeah. And not everyone likes that, and not everyone wants to be breathing that in. Totally understand. And a lot of folks will use carbon scrubbers. We actually have a carbon scrubber that I bought from a grow supply store up in Gun Barrel, which is not too far away. So that basically will absorb some of these VOCs and pull them out of the air so that you don't smell them. So the VOCs contribute to a lot of those smells, particularly the terpenes like myrcene. It's kind of that kind of dank, skunky sort of smell. So that's usually kind of the tried and true. Whether you're a home grower or on the commercial scale, you'll probably have some type of activated carbon sort of filter system. Even HEPA filtration can help with some of these larger particulates. So VOCs are gases, and particulate matter can be captured by HEPA filtration. So you can actually filter out some of the particles that may contain some of these VOCs and other things. A lot of cannabis growers will just not bring in fresh air or put out fresh air in their facility to kind of avoid some of these, neighbor complaints and things like that. That can be a problem though, because if you are not getting fresh air in, or you don't have an appropriate HVAC to really handle the capacity of these large grows, you can actually start enriching for things you don't want in your indoor environment, like mold spores and in airborne bacteria and things like that. And then I know here in Colorado, I've been to a couple of consumption lounges, and in order to get a license, you usually have to either have an outdoor section or you have to have pretty extensive. I didn't get a good chance to look at the filtration methods that they have, but they usually have to have some pretty industrial, HEPA grade filtration going on in those indoor environments where you're allowed to consume cannabis. But yeah, those are kind of the standard ones. I've also seen some advertisement for oxidizers to kind of, oxidize those VOCs and, change them so that they don't smell. The issue with that, and what I've seen with ozone in particular, so some places will do this not only to control VOCs, but to have low levels of ozone in their grow to hopefully control for certain airborne microbes. Ozone can actually react with VOCs and create all sorts of bad reaction products. Yeah, exactly. Ozone and see, this is something that the cannabis space is a little bit resistant to, because ozone is used a lot. I mean, you can buy an ozone generator off of Amazon. And so, they'll use ozone for all sorts of things. They'll pump it in into their grows, which is a worker safety issue too, if you're going up high, and they usually don't have ozone monitors in these spaces either. And so, if they're reacting with the VOCs in there, could create formaldehyde, could create other submicron particles, which are just harmful to breathe in no matter what. And and that's definitely an issue. So I'm not usually an advocate for those types of VOC kind of control methods.
And I would imagine there are ways to lessen that odor, but on the other hand, it's kind of catch-22. People want the odor, but are there ways to reduce the odor?
Well, the thing is cannabis is usually bred for the dankest, smelliest, flower that you can get. So there may be, I know that like as the environment warms, you're going to get more volatilization of these terpenes and things like that, but also plants like to grow in warmer temperatures. So it's kind of a cost benefit sort of situation where you want to have those VOCs present because that's a quality attribute of your product, but your neighbors might not like it. And if you don't have proper control methods, then it may end up being you may get called on by your neighbors. Yeah.
Well, actually, you can be called on by more people than that. Actually, in Pennsylvania, we were involved in a project where there was a hemp processing company that had come in from out of state, and I think they originated in Colorado, by the way. But in any event, they came here, and all they were doing was processing, and they were processing this in a very, very large building in which they were not the only tenant.
Oh,
And,
yeah.
okay, so what happened was there was contamination within the building of other people's products. Some of the companies, one of the tenants made radiators. That was really not an issue. The other, one of the other tenants was stored very, very high-end printing. Like boxes for high-end chocolates and candy and food and all kinds of stuff, and they wanted this stuff to be odor-free. And and what happened was this building, they had no containment, they had no carbon filtration. In order to dry it, they had brought in a large trailer-mounted desiccant dehumidifier. They were pumping hot, dry air into the building and exhausting it. And I mean, neighbors were upset for miles around.
Yeah.
I mean, this is a major, major issue. And people called OSHA, people called CVC, people called any organ, alcohol, firearms, tobacco, anyone that they could end up complaining to. And they ended up getting shut down, actually. There was so much outrage from local neighbors, and this plant was located kind of in a little gully, and you'd kind of have these inversions, and the odor would just kind of like stay there. And, this is out in Jeanette, PA at that time, Joe. So no, it was crazy. But what we found though is they did need to remediate some of the stuff that was printed, and we were able to determine, number one, that it had been contaminated, and we used these Tedlar bags, and we used Tedlar bags to grab these air samples, and we sent them to a special lab that kind of specialized in the cannabis area, and they were able to pick up all these different terpenes and identify them and so on and so forth. But in order to remediate, what we found worked very, very well is dilute chlorine dioxide. It worked really, really well, and they were able to sell their merchandise, and that worked out well. But
chlorine dioxide is also a common oxidant used in the cannabis space for sanitizing, both for like room turnovers,
right.
Both aqueous and gas. But again, you have to be careful about what you're going to expose your workers to and your plants to, because they're not going to like it either.
Right.
Like,
well, no, this is a processing. So, what happened was this treatment was made in the printing facility that actually printed it. But, the problem was how it got contaminated is they, would print, multiple months in advance of what they needed so the clients could have it. And they didn't have enough storage space. So they stored it in this more public warehouse, and kind of that's where the problems were.
Yep. And it also kind of sounds on point with cannabis, and I have a little less experience with hemp operators, but a lot of times they'll ask for forgiveness instead of permission.
Oh yeah.
You know what I mean?
Yeah.
And there's a lot of stuff that's done that's like on the kind of shady side, and maybe you shouldn't be doing it this way. Maybe you should probably control your process a little bit better. But that's not really been adopted in the cannabis space. Like, process control is not really a thing, and you get a lot of pushback from growers who already feel like they are overregulated as it is. And so there's a lot of room to grow there, and definitely I could see that as being like definitely going too far. with regard to not caring about the other operators in that building.
Well, there also weren't, I think, a lot of laws and regulations. The state kind of approves it, and they say yes, you can do it, but they don't have all the rules and regulations
Right.
either.
Yeah.
So I think from an enforcement state, I think the state was was somewhat negligent in not having it approved.
Better regulated.
Yeah.
All right, let's get into your paper, Bioaerosol Exposures and Respiratory Diseases in Cannabis Workers. I was reading back through the Bioaerosols book, and the statistic in there, and I don't have it in front of me, but I believe it was in one state they looked at respiratory disease in workers, in the workforce, and I think 10 out of 800 were related to cannabis, which really surprised me. Can you tell us a little bit more about what types of issues people working in these facilities have?
Yeah. So I've been into a couple dozen facilities in the US, Canada, and Australia. So I've talked to a lot of people in these spaces. I worked, for a year and a half directly in these spaces. So I know what people are exposed to. It really kind of depends. When it comes to respiratory exposure, on the cultivation side, you definitely see that folks will, like, I just spoke at the Cannabis Research Conference up in Fort Collins last month. And after I talked about this, a grower came up to me and she's like, whenever I'm in, she actually had an outdoor grow. And she's like, there's this one strain of cannabis. When I get anywhere near it while it's growing, I have a hard time breathing. My eyes start watering. I don't know what it is. And it's just this one strain. And I don't know what's going on. And I'm like, I have heard that so many times from so many cultivators. So there's something volatilizing in, in that cultivation side where you have the plants. It could be something chemical. It could be a chemical irritant that's bothering people. It could be biological from, handling the plants and disrupting them, and the plant detritus and the substrate that they're in. The fertigation or the watering system can also aerosolize water that contains microorganisms, creating these little droplets that could have things in there that could be potentially harmful. During harvest, the bioaerosol levels get really, really high because you're really disrupting the plants and moving them around. And there have been some studies, just a couple of studies looking at basically spore counts and plating for CFU counts of total yeast and mold. And like those levels basically reflect the microorganisms you see in water damaged buildings. So in these remediation sites after floods and things like that, it's on par with some of those sites. So clearly there's some type of potentially bioaerosol. A lot of these people on the cultivation side will also have contact issues. So they'll get rashes and things like that when they interact with the plant. And that is not very well understood what is going on there. And then on the manufacturing side, so when you're actually handling the dried plant material, milling that material, packaging it into pre-rolls or joints, there's exposure to mostly plant matter at that point, plus any microorganisms that were on that plant matter. And there's been a couple, again, surveys of these facilities. Again, there's like literally just maybe a half dozen papers really investigating these types of questions where they measured like LPS or endotoxin. In hemp processing, it was off the charts, like in the thousands of EU per gram. In the two studies that I've found in cannabis, it was below that. Of course, there's no OSHA limit for LPS exposure, but below the DCOS limit, which is, kind of more established in Europe, it was just below that level for like a 15-minute sample that was taken during grinding. So it's really not very well understood, but it appears that it's likely, maybe some of these viable pathogens could be in the air floating around. Those could cause problems. The microbial metabolites, we know well from other parallel industries, exposure to microbial bits and pieces can be harmful, whether it's endotoxin or LPS or different beta glucans or gosterol, maybe even potentially mycotoxins. We don't really know anything about that, to be honest, in the cannabis space. And then allergens. So those are protein allergens from molds and from the cannabis plant itself. So you can develop allergies towards cannabis, just like you can towards any other plant. Peanuts are plant derived. Pollen is plant derived. Plants are actually one of the biggest sources of allergens. And so you can develop allergies. And some of those people, so some of these studies that have been done, almost all of them on worker safety and trying to link that to some specific exposure in that environment, has been done in Washington State. So like that's where almost all the information is coming from. And some people do have, when you make like a cannabis slurry and you use it for skin prick tests, you can see that people do have an immune response to that cannabis slurry, but those slurries are not well characterized. We don't know exactly which allergens in that slurry is actually leading to that allergic response. Many people cite exposure to mold as the reason why they're having respiratory issues in these spaces. But again, follow-up on that has been very poorly characterized. And actually, a cannabis worker in Massachusetts died after exposure to cannabis dust in Massachusetts. And this came out, it was a report by the CDC at the end of last year, kind of going through all the nuances of that case. The worker did not have asthma before she started working at Trulieve, which was the cannabis manufacturer. Then she started developing asthma symptoms while she was there and actually had to go to the emergency room about a month or two before she had her fatal asthma attack. And OSHA came in and did an investigation, and they cited Trulieve for several citations. One of them was, you should warn your workers about the hazards of the inhalation hazards of cannabis dust. And they came back and said, there are no scientific studies investigating the inhalation hazards to cannabis dust. And so OSHA had to drop that charge. And so it's a huge gap in the cannabis space with hundreds of thousands of people working in this industry. I think the last report I saw by Leafly back in, I think it was 2022, was that there are over 400,000 people working in this space. And so many of them are being exposed to either, who knows, from a microbial metabolite side to mold allergens, to cannabis allergens, or other components we have not parsed out. And we just don't know what it is doing and why it is, why these environments are causing specifically these occupational asthma issues, but also other respiratory issues.
It's a very complex issue, and I think in the paper you had a figure there that kind of helped tie that all together. Grayson, can you put that up for us? Here we go. This just kind of tied it together for me. It's basically what you just said, but in a graphic that you can look at. Look at all the different things. I mean, nobody can test for all these things.
Well, it'd be great if there was someone doing this. I know that NIOSH is interested in working on characterizing some of these bioaerosols in these spaces. Like I said, most of the work that's been done in cannabis has really been looking at microbial populations, either through spore counts or CFUs or sequencing of DNA in those aerosol samples or collected dust samples. And that would really be focusing again on this top red bar, which is really looking at microorganisms that could cause infectious disease, maybe a link to allergens too. Again, allergens often aren't directly measured in the air. They're usually measured through a surrogate like counting spores. But you have all these other things that could potentially be harmful. Microbial breakdown products, bits and pieces of microbes, secreted products into the environment. Like endotoxin or gosterol, peptidoglycan, beta glucans, mycotoxins, and other bioactive agents. All of these are known to activate different parts of your immune system. And so they don't cause infectious disease, but they can cause or be linked to other types of more chronic respiratory diseases, especially after repeated exposures. And we see this a lot when we're studying agriculture dust exposure. So one of the co-authors on this paper is Dr. Tara Nordgren from CSU. And this is what her, scientific career has really focused on. She and I also, we both went to University of Nebraska Medical Center together. There's a lot of people in Nebraska and other agricultural dominant states that are exposed to agricultural dusts. And there's lots of stuff in those dusts, and they're also poorly characterized. But you end up seeing a lot of COPD, a lot of, these other types of lung diseases, and even exposure to these bioaerosols can be linked to neurodegenerative diseases. And so it's not just the lung that can be affected by that. It can be more systemic. And then on the blue bar here, the protein allergens, those again are a very specific type of immune response. This is an IgE mediated response. It's kind of your targeted sniper of your immune system, if you will. And that is it's going to be very specific, and you have to be exposed more than once to start developing these allergies. And that's also linked to asthma, as are the exposure to microbial metabolites. And if you have allergies, it can be severe. You can airborne allergens are one of the biggest triggers for asthma attacks. And so really, we don't really have a good idea of anything in the green or the blue bars here and how that's contributing to more chronic exposure and diseases in cannabis workers.
So I can imagine we have a pretty good industrial hygiene base within our audience, and I would imagine some of them have been called out and asked, okay, can you help us with exposure to bioaerosols or particulate in general, or et cetera? What do the growers currently sample for or test for, if anything, and what maybe should they be focusing on that they're not focusing on, right?
That is a great question.
Okay.
No, keep going. Do you have a follow-up?
Well, and I'm just wondering, like, if one of our industrial hygiene audience members gets called into a facility, where do they start?
Yeah. I think that this is something that needs to be bridged desperately between the cannabis industry and industrial hygienists. There's a big disconnect there. I've seen industrial hygienist reports on cannabis facilities, and growers will pay somebody to come in and, do kind of your standard spore traps and things like that. But then a grower doesn't really know what to do with that information. And the industrial hygienist will be like, well, of course you have, high PENASP levels because you're growing plants in an indoor space. And this is an issue. I think,
and there is no, PEL or TLV for this. What do we compare
there's
it to?
no PEL there's no permissible exposure limit to mold spores. There's no permissible exposure limit even to LPS for OSHA rules. There are exposure limits for dust when you look at cotton milling and wheat milling. But those levels are pretty egregious. You're talking about 5 mgs per cubic meter. That's pretty high when you think about how much particulate matter has to be in the air to reach that threshold. So I think that from both a worker safety and consumer safety perspective, we should do environmental monitoring the way that we do it in food. A lot of times you're looking for those most common pathogens that are going to be in the environment that can get on your product and pose a risk to consumers. So in food, you'll screen for, if you're selling cheeses and things that are going to be in cool, moist environments, you're going to look in your environment, in your manufacturing environment, for microorganisms like Listeria monocytogenes, which we've probably heard about the boar's head outbreak recently. And so I think that that's a really good place to start. But a lot of these, there's really no regulations or guidelines on this. There are some parallels in leafy greens that are mostly grown indoors. And a lot of it is kind of mostly focusing on those foodborne pathogens, E. coli, salmonella, things like that don't typically become airborne. They certainly can, but it's not like in the cannabis space where you're making a product that's going to be inhaled. And so you want to look at those inhalation hazards that could be in your environment, carry over onto your product, and then be a risk to consumers. So I think environmental monitoring for Total spores in your air is a good thing. Looking for specific pathogens like Aspergillus fumigatus is something that I think folks should start doing. It's not always it's like a lot of growers will push back on this issue because they say Aspergillus fumigatus is a ubiquitous organism. Well, it is, and so is other organisms that you have to test for in the food space. So, what you want to do is kind of target those high risk organisms like Aspergillus fumigatus that we know transfer through the air, and try to minimize their effects as much as possible. You might not, never see it in your facility, but if you can get it below like 1% of your hits, that is really good control. And so, sampling your air, sampling your product, making sure that you don't have an environment where these microorganisms can really thrive, where you have poor ventilation, poor sanitation, which is rampant in the cannabis space. There is no established good sanitation practices for cannabis. Just good handling.
Could you say that again?
There are no established good sanitation practices for the cannabis industry. And the idea, like in food manufacturing, you have a whole sanitation team a lot of times where you clean before you shift, you clean at the end of your shift. That is not typically done in the cannabis space. And without FDA oversight, there's no enforcement that good sanitation is followed.
There we go again, back to the federal. Okay.
Yeah.
Okay. Cliff, I know you had a follow-up.
Well, yeah. One of the things, and you were talking about food and food management and so on and so forth. One of the things that's used a lot in the food industry is ATP. They use it on different surfaces and so on and so forth. But it would seem to me that ATP is going to go off the scale in this environment. So most likely it would not be appropriate then, because there's just too much.
I think ATP meters are awesome. I love them. I think that they're a good verification tool. So I think that they're most appropriate on the manufacturing side. So after you've harvested, you've gone through dry cure, now you want to make sure that all your equipment that's going to be touching that product, all your product touching surfaces, all your equipment, everything that's going to interact with that product is clean, it's sanitized, and it passes ATP testing. And that, I think, is where you can basically follow a lot of other manufacturing rules and guidelines for food and other kind of parallel products. And even brewers, my husband's a brewer for years, and they used ATP meters. So they're not necessarily required to, but they do it because it's good quality control, quality assurance. Right. So I do think that is a good space for that. Again, as a validation or as a verification step, I'll be very clear, because in food, validation and verification are two different things. As a verification step, yes, I think that ATP meters and swabs can be very useful.
Oh, thank you.
We're running a little over, but hopefully you can stick around for a minute. I got to ask one more question here, and that is, why wouldn't a manufacturer just use the precautionary principle and give their people the proper PPE, have the proper ventilation and filtration? Have you seen that in the industry?
I was really spoiled when I was working at Columbia Care in New York. I didn't realize how good I had it. Like we had, so my boss, the operations manager, he was an old Kodak guy, and his dad, I think, was like the state OSHA inspector, and he really cared about worker safety. And so we had a respirator program in place. We would have an industrial hygienist come in and sample the air for all sorts of things. We'd get fit tested, we get our EKGs, and then we had a giant PAPR that if you, for some reason didn't want to wear a mask, you could wear your spacesuit, while you're grinding flour, things like that. When I go to other cannabis facilities, those basic worker safety and good ventilation, the hierarchy of controls, is just not really understood by the industry. I've definitely come across growers who think that OSHA regulations don't apply to them because they're still federally illegal. And so that is just conceptually, it's kind of a disconnect with some of the leadership in these companies who are really focused on making a profit as quickly as possible, and maybe don't always think that it's a plant they're growing and that there are people in there that have to do it. And so, yeah, it's just kind of a lot of oversight, I would say. Like, people aren't really putting in the proper ventilation, having, experts in HVAC come in and properly assess their setup, having industrial hygienists come in and sample their air, or even, just doing basic quality control and assurance in-house is kind of unheard of. Most of the time, they'll send it out for third-party testing labs. So there are labs that are dedicated to test final product on cannabis, but sometimes they will, like growers and manufacturers will send them, they'll ask them to come in and do like settle plates. So they'll put out auger plates and but like a lot of times there's still a disconnect there because these are compliance testing companies. They're not environmental, testing companies. And so there's a disconnect there. And then a lot of times the growers will just blame the third-party testing labs for not knowing what they're doing and not take on the onus of being a manufacturer, and that responsibility should be on them. So yeah, there's a lot of disconnect.
What can we
Global-
learn?
Oh.
Other people who work with indoor environments, what can we learn from the cannabis industry?
I mean, there's a lot of innovation in cannabis. Even though I'm here kind of like being like wagging my finger a little bit, I really do think that cannabis growers are very adaptable. They're very smart. They are very practical, and they do a lot with very little. And so I've seen like some really cool setups for different types of grows. From you have your traditional grows, but then you have like aquaponics and hydroponic and aeroponic, where you just spritz water and nutrients on the roots. So I really think that there's a lot that the cannabis space can offer. It's one of the biggest indoor, it's called controlled environment agriculture, or CEA. It's one of the biggest, most high profitable CEA environments out there. So I think we really should be looking to the cannabis space to be leaders in this industry, leaders in the CEA space, and really setting the stage for every other, high value crop that can be grown indoors. And I think that if we give them the resources and we give them the tools, most of the growers I know would run with it. They just need to be pointed in the right direction and have kind of that context. Okay, now I have this industrial, hygienist report on the number of spores in my air. What do I do with it? How do I transfer this into something meaningful? And I think that that's where we just really need to build more bridges between, the industrial hygienists, the academics, the scientists, and the cultivators and manufacturers, because right now everyone is really siloed.
Very similar in other industries. Cliff, do you have any final questions or thoughts?
I did. It seemed to me that this is where you need a trade association of growers or whatever.
Good point.
And I'm sure there probably are trade associations, but I'm just not sure they're focused perhaps where they need to focus.
I think you're probably right on that.
But yeah, well, there are two. I guess the second thing is, globally, where would you rate the United States in terms of this industry? Because you talked about Canada, you talked about Australia, Europe. I mean, it would seem to me that maybe we were later comers to this industry than perhaps Europe, and so on and so forth. Are they doing a better job over there? Do they have it more together?
I mean, hands down, in my experience, the US has the best cannabis that you can get. Now, the best is like usually measured again by those sensory aspects. How does it smell? What does it look like? The safest, I think it's probably on par with other countries. However, most of those other countries have some type of federal oversight. And so at least it's the same across all, provinces or states or whatever. And here it's more piecemeal. And so there's just disconnect, you can't ship across state lines. So there's a lot of issues with that. And again, I mentioned this before, but there's this whole process of taking contaminated, often visibly moldy cannabis and recovering it through this process of remediation. Some people call it a microbial control step, but in food manufacturing, you can't take adulterated product and recover it through decontamination methods and sell it to consumers. I can't take spoiled leafy greens, zap it with irradiation, and sell it to people. You can't do that, but you can in cannabis. You can do it in most states, not all states in the US. You can do it in Canada, and you can do it in Australia. And so I think that that's a major issue that needs to bring it. We really need to bring attention to this. The European Medicines Agency has recommended that cannabis be tested before it's irradiated or treated with some type of decontamination method to make sure you're not just trying to recover, failed product. And, other organizations like the United States Pharmacopoeia have said that this process of remediation should not be allowed, but states still allow it, even though they follow USP on other regulations. They don't follow this particular regulation. I think labeling of those products is really important and must be disclosed to consumers if a product has been treated, even so people get around this a lot, especially here in Colorado and other states. They'll treat before they send it out for third-party testing, so they never have to declare to the state or anyone that it's been treated. You only have to declare it after it's failed, and then you recover it through remediation. And then you have to declare it to your state, but you don't have to put it on the label or tell consumers that their product would have failed if they hadn't treated it with some type of reactive oxygen species, ionizing radiation, radio frequency, things like that. And so I think that that basic quality assurance, and again, controlling your process so those microbes don't get out of control, either because your plants were infected with something and you just were like, whatever, it's not a human pathogen, so who cares if my plant's infected? I'm going to harvest that and recover every gram of flower that I can. Or in your post-harvest space, a lot of post-harvest foliage microorganisms that are not unique to cannabis, they're in other agricultural commodities, really controlling that environment and testing and just putting in some basic process control would really help. And that, as far as I know, Canada has some good practices for medicinal cannabis, but they can still recover through the process of remediation. So I think that that's poor practice, but we're all learning as we go. So hopefully this is an issue that it starts getting some more attention from a consumer safety perspective. Because as I mentioned in that figure, yeah, you have microbial pathogens, but then you have all their bits and pieces and allergens and things that are likely going to survive. And we know that some of these things do carry over into smoke. So then you're breathing that in. So yeah, I think that that's a major issue that from a consumer safety perspective needs to be addressed everywhere.
Well, and that's a whole nother area that needs to be looked at from the perspective of worker safety. Who are the people doing this decontamination? Or whatever what are they exposed to? My goodness.
Absolutely.
Oh my. Well, before we go, is there anything you'd like to add? Anything we missed?
No, I think this was great. It was really good questions. I appreciate the time and getting your guys' perspective on this. Thanks so much for inviting me.
Our thanks to Dr. Tess Eidem. Fantastic. Great show. My co-host, the Z-Man Cliff Zlotnick, our engineer, Grayson Gone Fish and Fisher, our sponsors, and most importantly, our loyal audience. Next week, we're coming back with one of our most popular guests, Dr. Joe Spurgeon. We're going to talk about writing a mold report, the basics. Get back to the basics, everybody. So we'll be back next Friday at noon with the next episode of IAQ Radio Plus.
For IAQ Radio, I'm Spike Reed saying thanks for listening.